Hello and welcome to the lecture. So, in the previous lecture, we have studied about how we can synthesize amino acids. Or we have seen in the previous lecture how preparation of amino acids is being done. And we have studied two methods in the last lecture, the synthesis of amino acids. One was by doing aminolysis, and another method that we have studied, it was Stroker synthesis. Now, in today's lecture, we will be studying about another method by which amino acids are being synthesized, and this method it is called as Gabriel Phthalimide synthesis. Gabriel Phthalimide synthesis. So, what this is all about? First of all, let us see what types of reagents are being used in this type of synthesis. So, the reagents that are used, it is potassium hydroxide, and another reagent is alkyl halide. Now let us look at its preparation. Now the preparation we synthesize this from phthalimide. Like this is our phthalimide, and here there is nitrogen here, and we had two carbonyl groups. So this is our phthalimide. Now this phthalimide it is treated with potassium hydroxide. Now we know that this potassium hydroxide it is basic in nature. Now if this is basic in nature, what this will do? This will remove this hydrogen. Now when this will remove this hydrogen, what will happen? We are getting this type of species. This type of species will be introduced in phthalimide. Here we have, this is carbonyl group here, and here we have positively, this is negative charge, and this type of species is being generated. Now in the next step, what happens? This is treated with this Cl. Here let us write down here. Here we have chlorine, C double bond O, and we have here R dash species. And there happens what type of reaction? There occurs SN2 type of synthesis. SN2 type of reaction occurs, and this chlorine it gets removed. Now when it gets removed, we are getting this type of product. Here, since there occurs SN2 type of reaction, we are getting CH2 here, C double bond O, and here we have OR. We have here OR, and this is CH2. Now in the next step, what happens? Hydrolysis occurs. Now when hydrolysis occurs, this all is being removed, and we get this type of species. Dicarboxylic acid type of species, and there is formation of amino acid. CH2 here, we have C double bond O, and we have here OH. OH So this is plus there is a removal of an alcoholic group. So this is how the amino acids are being generated. In case of amino acids are being here, this CH this we have actually here one CH, and there can be different R groups. So this type of synthesis, this is called as Gabriel Phthalimide synthesis. So the starting material here, it is phthalimide. Phthalimide, it is treated with potassium hydroxide. Now since this hydrogen, it is basic, this hydrogen, it is acidic in nature, and here we have basic species. So this basic species, what it does, it removes this proton, and this results in the formation of this type this type of species. Now in the next step, SN2 type of reaction occurs, and here this attacks on this carbon and chlorine is being removed, and we are getting this intermediate, this type of intermediate. And in the next step, when we do hydrolysis of this intermediate, we are getting amino acids. So this is how amino acids are being generated using Gabriel Phthalimide synthesis. Now we are studying, so this was all about how amino acids are being synthesized. Now we will see, in case of amino acids, there are peptide type of linkages. So we will see dipeptide and peptide linkage. Dipeptide and peptide linkage. Now how these dipeptides and peptide linkage are being generated. Now actually, when two amino acids like amino acid 1 and amino acid 2, they undergo dehydration. They undergo dehydration. That is, what is dehydration? There is loss of water. Then there is formation of a special type of linkage, and this special type of linkage, it is called as peptide linkage. Now let's understand this by taking an example. Suppose we have amino acid like this. We have, this is one amino acid, and we have here another amino acid.